Hello, my name is Cassandra Thayer, and I'll be reporting on horseshoe crabs and the ethical implications of harvesting their blood for medical usage. Horseshoe crabs are extraordinary creatures that have been around for thousands of years. They are essentially living fossils. Their blood contains amoebocytes, which have coagulogin in them. In 1956, scientist Fred Bang discovered that when he injected bacteria from seawater directly into the horseshoe crab's blood, it clotted around the bacteria, creating stringy masses. Bang suspected that this was an intentional evolutionary trait and developed the Limulus amoebocyte lysate test, more commonly known as LAL. Probably because it's very difficult to say. This test is utilized by the medical community to test vaccines, injectable medications, and other sterile pharmaceuticals. Each year, pharmaceutical companies and their counterparts harvest these creatures and bleed them for our benefit. Unfortunately, over time, this harvest has put a significant dent into the population of the species. Though scientists report only a 10% to a 30% mortality rate, studies have shown that wild populations are in danger. In 1990, scientists estimated the population to be at about 1.24 million. Just over a decade later, the numbers had decreased to 33,500. In 2019, there was a slight increase, but not very significant at all. Additionally, horseshoe crabs are considered a keystone species, which means that other species depend on them for survival. Migratory birds, such as the ruddy turnstone, pictured here, make the journey to Delaware Bay to feast on horseshoe crab eggs by the thousands. With a lesser population of horseshoe crabs, in turn, the number of birds have dwindled as well. Fortunately, programs have been established to help mitigate the population decline. One program, aimed at rescuing stranded horseshoe crabs, has begun to make an impact on New Jersey beaches. Return the Favor aims to not only rescue horseshoe crabs that are either stranded or flipped over, but to increase awareness of horseshoe crabs, shorebirds, and the management of both. Additionally, they collect data and information on stranded crabs, the potential hazards around them, and they will tag crabs to aid in management and restoration efforts in the Delaware Bay area. Though they are a small organization, no effort is too small for conservation. There have also been harvest limitations in some states as well. In Massachusetts, the limit is 6 unless you would like to apply for a regulated fishing permit. This permit would mean that the harvester would have to report their catches and their mortalities to the local government for research and tracking purposes. Additionally, in New Jersey, there was a moratorium placed on harvesting since 2008. Unfortunately, data from the Wellfleet Bay Wildlife Sanctuary shows that these efforts have not been able to substantially increase wild populations of the horseshoe crab. One method that I propose that would be more ethical than taking a horseshoe crab's blood would be a widespread use of recombinant factor C, also known as RFC. RFC is a synthetic version of the components in horseshoe crab blood and would eliminate the need to bleed these animals. Currently, it's widely used in Europe but the United States has yet to adopt the practice fully. The FDA does not feel as though it is safe enough to use for vaccines and other medical programs. If we and many other countries were able to adopt this practice instead of using actual horseshoe crab blood, populations could potentially be restored, and on top of it, the medical community would not be reliant upon an animal for the vaccines. Not only are horseshoe crab populations in trouble, but if the horseshoe crab disappears, then the medical community would no longer have a way to test the vaccines and other sterile medical medications and pharmaceuticals that we currently use. That would honestly be a win-win for everyone. I hope that you have enjoyed my presentation on the horseshoe crab and their crazy blue blood. Thank you.